strategy for pure and applied physics. Chart and I have been involved in this process of identifying physics in Africa, and especially biophysics in Africa. And we've written an article which I've given a link to, or I've given a link to my slideshow, which is based on the article. And this article is going to be presented as a component of a complete analysis of physics in Africa to UNESCO sometime later this year, probably December. And our plan is to try and actually attract attention to physics in Africa, and our obviously interest is in biophysics. And as a component of this, we're wanting to get a database of all biophysics-related equipment in Africa and to actually identify the viability of doing experimental biophysics in Africa and find out whether there's the capacity to make proteins and DNA and all these things at any number of sites in Africa, and to find out where this talent is, and then identify the people associated with all of these facilities, so that we can have a baseline for getting together some sort of biophysics community in Africa. And so I'm going to give you the link in the first place, and then during Yassine's talk, I'm going to give the link to the database. And the database at the moment concentrates on structural biology, but it only has South African entries. There are no entries from anywhere else in Africa. So I don't know whether it's true that in fact there are no electron microscopes, NMR machines, X-ray diffractometers, or protein preparation resources anywhere else in Africa. I can't believe that to be the case, but I'd like to appeal to people for help in identifying those. And okay, so with that, I'm going to go on to introducing Mohammed. Mohammed's going to talk to us about his recent work on the HIV protease. I'm sorry, Yassine is going to talk to us about his recent work on the HIV protease. Yassine is a Witsi. He's been at the University of Witwatersrand forever, and he leads the protein structure function program at WITS. And he's done a huge amount of work in propagating structural biology in South Africa. And I think that the survival and success of structural biology in South Africa is largely dependent on the efforts that he's made. And he's kept the subject going, and he's educated the majority of people that are actually active in South Africa at the moment. So Yassine is a force in our structural biology community, and he's going to talk to us about cryptic sites in the HIV protease. I never thought of the existence of these cryptic sites, and I read from his abstract that they're extremely interesting, and and potentially a new kind of drug target. So with that, over to you, Yassine. Please go ahead.
Thank you very much, Trevor, for that very kind and generous introduction. Let me see if I can do this. I'm going to hopefully turn on my video.
Okay, your face is not in the middle.
Okay, is that fine?
That's better, yes.
Okay, so I think that's all that I need to do, but I think I disappeared again.
When you share your slides,
yeah.
but then there'll be a little inset with your face.
Who can share? I'll say all participants, is that right?
Yes.
Okay, and who can start sharing when someone else is sharing? I'll just say all participants again. hopefully.
Okay, so
That's
yeah,
fine. Okay.
there you go.
There we go.
If you
And
go to full screen, you'll be fine.
is that okay?
Yes.
And I'll just want to see if I can get a pointer. Okay. Great. Thank you. Thank you very much, Trevor. And I think I also need to thank Chart um, and the organizing committee for inviting me. I think the talk was supposed to be scheduled a bit earlier this year, but uh, Trevor informed me that he needed to move some things around. And so I was quite happy to do it at this time because a few weeks ago, I think it would have been very difficult to give this talk based on all the teaching and lecturing and marking, etc. But I've had a bit of time to at least put a talk together. So thank you once again, and thanks to everybody for joining in. Um, Trevor, is it okay if I don't? I don't know. Are you able to see me? Because all I can see is my screen.
Yes, we can see you. We
Okay,
can see
brilliant.
you and we can see the screen.
Okay, lovely. So today I'd like to talk about um, these so-called cryptic sites or hidden sites. And they're not very well known or well characterized. And this was work that was actually done by my PhD student, Dr. Dean Sherry. So Dean was a phenomenal student. I really enjoyed having him in my lab. I lectured to him as an undergrad student, and then he came all the way through, an honors, masters, and PhD. And currently, he is working in the private sector for a company in Cape Town. And I know he's also doing some amazing work for them there. So today I'd like to talk about cryptic sites, and I've titled this Cryptic No More because we've managed to uncover this potential site. And I think we can regard this as a novel anesthetic site in HIV protease, specifically the cantilever region. And I'll explain a bit about protease structure and where this region actually occurs. So on to just a very brief overview. I want to actually just share with you, why this research? Why do we actually do research into HIV protease? And then maybe just look at some of the structural features of this enzyme, and then how we can go about probing or searching for these cryptic or hidden pockets, and some of the tools that we've used to uncover these cryptic pockets. And I'm going to talk a bit about molecular dynamic simulations, which I think everybody knows about, but something that's not really that well known or people haven't really used it that much, and that's mixed solvent molecular dynamic simulations. I'll touch a bit on docking and also tools that we can use for structure-based pocket detection and fragment-based virtual screening, and then I'll conclude and also uh, mention some acknowledgements. So why this research? Well, as you know, last year, 1st of December 2024, or the 1st of December every year, the world celebrates World AIDS Day. And last year we had the theme Equal Rights, Equal Care. And this is very important because we need to ensure that, especially the underprivileged, who are largely affected by HIV and AIDS, that they actually have access to health care and the correct and proper health care so that one could actually protect the public and also, secure their human rights. Um, so we actually have various treatment strategies, protease inhibitors, reverse transcriptase inhibitors, etc. However, to date, there's no real effective cure. Okay. And there's also no vaccine available to actually treat HIV and manage the disease effectively, HIV/AIDS. So there are a range of drugs that are currently available, from protease inhibitors, non-reverse transcriptase inhibitors, to inhibitors for integrase, as well as fusion inhibitors, etc., etc. So there are really, there's a whole host. There are many targets in HIV that have been identified, and some of these drugs have actually been used quite successfully to manage the disease and the spread of the virus. I think maybe of recent interest is lenacapavir, which is a P24 or capsid inhibitor. This one is quite interesting because patients only receive a twice yearly long-acting injectable. It's used as PrEP or pre-exposure prophylaxis, and it's been found to be very effective in preventing HIV. So this actually has shown some very good promise. And I think there'll still be, lots of progress that needs to be made before we can actually say that we can see an end to the AIDS epidemic. So it's still a long journey, and I think lots of research still needs to be done. So onto the HIV protease, let me introduce this enzyme that I've been busy with for several years. And I remember some time back, Trevor and I even chatted about HIV protease. So it's a homodimeric enzyme composed of two identical monomers. Shown here on the left is one monomer, and on the right, where I'm indicating, that's the other monomer. So these two monomers dimerize to form a homodimer. And when the enzyme dimerizes, there's the formation of an active site, which is shown here in gray, sort of in the center. And there are some very important structural features. So one of those features at the top of the enzyme, if we can call it that, are these flap regions, two flaps contributed by each monomer. And to the right over here in green is shown as a hinge region. And below that, we have a fulcrum composed of these two beta strands over there. And we also have another set of beta strands over here, and that's designated the cantilever region. And at the base of this enzyme, that's when we have the sort of interdigitation of the NNC terminal ends of the protein, and this is responsible for the dimer formation. What I've indicated on the right are the residue numberings for the flaps, so 46 to 54, the hinge 35 to 42, 57 to 61, cantilever 62 to 75, and the fulcrum 10 to 23. It's not really necessary to remember these numbers, but I think what's important is that I'm going to focus on this cantilever region quite a bit, which is, let's say, indicated by the region around here, and also in blue with these beta strands over here, and also the flap region. But I will touch on these as I go along. So what's important to note here is that the HIV protease, like many other proteins, exhibit elasticity. Now, I know in the literature, people have actually said that proteins are plastic. I actually prefer to think of them as elastic because I think if one also speaks to a physicist, there's a big difference between plastic behavior and elastic behavior. So if we think of proteins as being elastic, that means that they're actually capable of a dynamic and very flexible behavior without necessarily being um, made to denature. Okay, so they can expand and contract, and various motions are available to them in terms of their functionality and their stability. And HIV protease is no stranger to this, because On the left is a diagram showing you the HIV protease in a so-called closed conformation, and that is usually measured either from a glycine residue on the flap, sometimes an isoleucine, all the way to an aspartic acid residue in the active site. So when this distance is sort of around 9 to 11 angstrom, this is regarded as a closed conformation. When these flaps tend to open up slightly, you can see the distance increases between these two residues, one on the flap and one at the active site. And this is regarded as a semi-open conformation. And that's around sort of 15 angstrom. And if the protease exists in an open conformation, then the distance increases all the way to about 20 angstrom or even greater. So the protease is actually capable of this kind of elastic behavior, and this is very important for functionality. We'll just keep this in mind as I move along. So again, showing you the diagram on the right that I discussed previously, but here what I want to actually show you now with the surface representation with the secondary structural elements sort of also in the background are the existence of some potential so-called pockets. So at the top of the enzyme, close to where the flap is, that's referred to as the flap top pocket. This region in the center is called the active site. That's also where the natural substrate binds, so the protease can cleave the peptide bonds, generating proteins that are required for viral maturation and later on infection. But it's also the site where all the current antiretroviral protease inhibitors bind. So they all bind to the site, and that's termed the active site. The site to the right here of this fulcrum region has been described as an exosite, and at the bottom, at the base, which is where the cantilever is, this is what is termed the cantilever site. So there are these several sites, and but currently all the drugs that are designed focus on this active site, and there's nothing that actually looks at these other potential sites in an attempt to try and destabilize the enzyme or reduce its enzyme catalytic efficiency. So this is just an introduction to some important sites of interest. What I wanted to show you here is that the HIV protease has 99 amino acids per monomer. So this is quite scary because basically more than 60% of the protease can actually undergo mutations. Not all at once, but certainly certain combinations or individual mutations can occur. And some mutations occur within the active site, those shown in red spheres, and they can actually occur at distal sites far away from the active site. And these are just this is a mapping of the amino acids that can undergo mutation. And these mutations, quite worryingly, still allow the enzyme to be stable and folded, and it functions quite well when it has to cleave the natural substrate. But when the drugs bind to this active site, they actually bind with reduced efficacy, with reduced affinity, and that sort of presents a problem because the viruses can then develop an escape mechanism, or there's some form of drug-induced resistance that can occur. So this, therefore, is a very tricky, a very difficult enzyme to work with, especially with regard to drug design, because one needs to take into account, these potential mutations and how that may actually affect the drugs. So this is the kind of enzyme that we've been dealing with over the years. What I'm just showing you here is the type of flexibility that is available to the protease and why I actually say it's quite elastic in terms of its behavior. Shown on the left in the blue structure, the blue ribbon structure, is the wild type protease with these active sites, with the flaps in an open conformation. And this happens when there's no drug or no substrate bound. And I think what you'll notice immediately is that although the flaps are open, both flaps from each monomer don't necessarily open to the same degree. And this is a feature we've just noticed in these proteases. But we've also worked on a protease from a drug-naive infant that exhibited an N37 to T mutation, and the upward arrow V indicates an insertion. So the protease in this infant exhibited 100 amino acids per monomer as opposed to the original 99. And we've done quite a bit of work on this, and molecular dynamic simulations have actually shown that the flaps can actually open to a much larger degree compared to the wild type. And this obviously has an impact on drug binding as well as potentially the catalytic function of the enzyme. So flap and hinge region dynamics, and this is the hinge. Okay, these hinges are actually necessary because they actually allow the flaps to open and close. So you can't just look at the flap without considering the hinge region as well. So what we know is that these mutations, and we've seen several of these mutations now, they do in fact affect flap region dynamics and also the hinge, whereas the wild type shows decreased flap dynamics. So onto the main part of today's talk. I want to just sort of give you a brief overview of the methods that we used without going into too much detail. So yes, we did molecular dynamic simulations. And for that, we obviously accessed protease structures from the PDB. And this here is the PDB structure ID, PDB ID rather, of the HIV protease that belongs to the South African subtype C protease. And I think Trevor would also recognize that PDB code because that was the first South African subtype C structure that was published a few years ago by my PhD student, Prevan Naika. And this structure has actually been used many times by many groups, to represent the so-called subtype C structure, especially, a representative structure of what the protease would look like in sub-Saharan Africa or South Africa. We've also used the SIV or simian immunodeficiency virus as another form of retroviral protease, Rous sarcoma virus, feline immunodeficiency virus, human T-cell leukemia virus, as well as Mason-Pfizer monkey virus. So you'll see in a moment why we've taken representative proteases and why we wanted to see if our method and our rationale actually, was solid. All structures that were generated in this talk were generated using PyMol. We generated the dimeric form if only the monomeric form was available. I will produce some results later. We generated a disulfide bond, and that was done using disulfide by design. I'm not going to go through these. These are just the parameters that we set in order to define the disulfide bond between a cysteine residue 72 and 92 from each monomer in the protease. And then we use GROMACS for molecular dynamic simulations. I'm not going to go through the details, but we've used these high power computers. We also took care of any protonation states of ionizable residues. We used a certain solvation model called TIP3P because these simulations were done in a solvent environment, a water environment defined by a certain cubic box. Simulations were performed for about 1,000 nanoseconds, which I think is quite impressive, given that one needs a significant amount of computing power to actually do this. So 1,000 nanoseconds or 1 microsecond is very decent in these days. When we just started off with molecular dynamics simulations, I recall we could only go up to about 20 nanoseconds, and in order to generate sufficient data to view the molecular dynamics simulations, it took us about 1 week to actually do this. So we've now got enough computing power to do these very comfortably, over a few hours. We used a particular force field. I think in any molecular dynamics simulations work, it's always important to define the force field. And there's several types of force field, but in terms of the software that we used, it was the AMBER 99 SB force field. We also took care of long-range electrostatic interactions using a particle mesh Ewald method. Also looked at short-range electrostatic and van der Waals interactions with a cutoff of about 1 nanometer. And then, yeah, I don't think people you really use mixed solvent molecular dynamics simulations that much. So I thought I'd just define this very briefly. So it's a type of molecular dynamics simulation where one takes a biomolecule, which can either be a protein, it can be a nucleic acid, and then you simulate that dynamic simulation, but you include both water as well as another solvent molecule. So when one does molecular dynamics simulations in a solvent environment, that's only with water. But the minute you include another molecule, then this is now termed mixed solvent because you have water as well as a small organic molecule. Mixed solvents are very good, or they are capable of identifying or locating these so-called hotspots or druggable pockets. And these are generally on the surface. Some, depending on the type of molecule, you might be able to get into the interior of a protein. But for the most part, these are surface-based kind of simulations. What's important is that when you do this, you need to understand what the free energy minima calculations are, and also how long each of these probes spend, how much time they actually spend in these binding sites. So these are termed residence times, and binding free energy as well as residence times, these help to identify these hotspots. And what we're actually after are binding energy values that are as low as possible, and residence times that are as high or as long as possible. And that sort of would give us a bit more confidence in the fact that we can say this molecule is potentially binding to an important site. So if anybody wants to have a look at mixed solvent molecular dynamics simulations, you can look at this paper by Martinez Rosell, published in 2020, and the details are shown here. I'm not going to go through all the finer details, but I just wanted to show you what the workflow was for mixed solvent molecular dynamics simulations. First, one has to parameterize your molecule. You look at the charge. You take care of any rotamers by making sure that they're in the correct stereochemical configuration, that there are no clashes, and you need to obviously optimize your topology. Then one can perform the simulations. Usually you start with an apoprotease or your enzyme in the absence of a ligand or a drug. And then you can also prepare your protein by looking at protonation and relaxation, so that the configuration of the enzyme is in its lowest energy state. You can then perform your simulation in explicit solvent. You can define your cubic box. We centered a protein at about 6 angstrom from the edges. You then add your probe molecules randomly, or one at a time, until the probe concentration is about 0.1 molar. that seems to be optimal for binding. And you can then run your simulations for about 200 nanoseconds using a specific force field. You can then identify your hotspots and then rank them according to their resonance times, and also in terms of how long they spend there, and also in terms of the energies. Remember, water is also part of this simulation, so you need to also look at any occupancy by water as a probe. Once you've done that, you can then identify your location, or where these probes remain bound, and then try and study the molecular features, or the chemical and molecular features of these probes. So when you probe the surface for these pockets, as I said, it's very important to get the lowest free energy minima, also looking at the amount of time that these probes spend in the hotspots. And what's very important is to use several probes and not a single probe, because several probes with slightly differing chemical properties might actually help you uncover some sites that you wouldn't be able to be uncovered if you were only using a single probe. So in our study, we used five probes. We used benzene, imidazole, isopropanol, phenol, as well as acetone. Um, and we found that these five molecules actually frequently sampled these so-called hotspot regions. Um, and the pocket, or the region that was sampled most often by these probes, was a pocket near the cantilever tip, and this is what we term the cryptic site. So, well, I think that this is also very important. This cryptic pocket, when one looks at, or when one studies the APO enzyme, you won't see the existence of this pocket. It doesn't show up. Even when a protease inhibitor is bound at the active site, the cryptic pocket still does not show up. Okay, so in the APO form, as well as in a drug complexed form, the pocket still remains hidden, and therefore behaves in a very cryptic nature. It's only when we did mixed solvent molecular dynamic simulations using these simple molecules that this cryptic pocket actually revealed itself. And I thought that was really quite cool. Water-only simulations were also unable to induce any opening of the cryptic pocket. So even just simulating the event using water alone, because all these enzymes are cytosolic, and so they would exist in a water environment, that cryptic pocket does not reveal itself even in the presence of water alone. We then also went on to do some molecular docking. For the molecular docking, Dean looked at a chemical library from Life Chemicals General Fragment Library, and we screened about 65,500 fragments using virtual screening methods. And we then applied certain restrictions when we ranked the top best molecules. So we filtered many molecules out, and we selected about 10 molecules that met certain very stringent criteria. And we looked at that further in terms of whether these molecules have the potential to bind the cryptic pocket. We then conducted docking procedures. We also extracted the HIV protease structure from our mixed solvent simulation models using benzene and a co-solvent to actually use these cryptic pockets in combination with the fragment library to see if they're actually bound. The structure we used was prepared using this software prepare receptor. We removed all the water molecules, added hydrogen atoms, etc., in order to produce the protease for further study. The cryptic pocket was then identified, and we also used it for binding or docking studies. And we set a certain exhaustiveness level. We set it to 8 to balance the computational efficiency. And we also wanted to generate multiple binding poses for each fragment, just to see if the fragments actually bound in the same orientation or in slightly different orientations in that cryptic pocket. So structure-based pocket detection. So this is software that one can also use to try and get some more information about the actual pocket. Once you've identified the pocket, if you need to study the pocket even more, you can use these free tools. And we used 4 of them. We've used FPocket, DeepSight, CryptoSight, and DarkSight. You can also use another tool called PocDrug. This allows you to assign geometry when these molecules actually bind to these sites. FPocket, this method uses a binding site prediction and also a druggability score calculation. DeepSight is a machine learning tool. It predicts protein ligand binding sites, and it seems to be the tool of choice because it apparently shows that it has outperformed many so-called state-of-the-art methodologies to date. DeepSight is another one. It actually helps you understand protein 3D structure, hydrogen bond acceptors and donors, any information about ionizable groups, whether they are protonated or deprotonated. You can also get an understanding of the hydrophobic residues and hydrophobic interactions. And it actually utilizes a deep convolutional neural network in order to predict ligand binding. A bit of a tongue twister. DarkSight is another one. It also helps you to predict druggability on these pockets. And again, it looks at several chemical and geometrical characteristics. And then we used a support vector machine to look at druggability predictions and then score these in terms of trying to identify, the best molecule and also the best druggable pocket. So the probes that we used, I'll just put them up very quickly, and I'm sure you'll all recognize them. So that's the structure of benzene. This here is the structure of imidazole, as well as a phenol group. So we started introducing a bit of sort of a polar character to this very hydrophobic so-called non-polar molecule. And then we have isopropanol as well as acetone. And that would just be a representation of a water molecule. So these are essentially the probes that we use. Very simple probes, very simple molecules. But I think what it uncovered for us was actually amazing. Okay, it was really an eye opener. So one doesn't have to use very complex molecules in order to get a protein to reveal its secrets. What I'd like to show you, and I really hope this works, are 2 molecular dynamic simulations that we've done. The simulation on the left is just for the apoform of the protein, and the simulation on the right is a simulation with benzene only. And we conducted each of these for 1,000 nanoseconds, but at some point, I won't let the entire simulation run because at some point, there's no real observable or noticeable conformational change in each protein. So let's see if this actually works. Oh, there we go. Great. Okay. So what we notice here on the left is a molecular dynamic simulation of the apoprotein. And again, you'll notice the active site, the 2 monomers. And here you have the flap regions which are actually closed. The one on the right, again, shows you the simulation with the benzene molecule. And perhaps what I should just do, let me see. You'll notice the benzene molecule samples some of the active site. It goes into another site over here. It spends a bit of time, and then it goes into a site over there at the top of the flap or the flap pocket, until eventually it sits in this region, and the residence time and obviously the dissociation kinetics become very, very slow. Okay, and it spends the majority of the time in this region over here for the rest of the simulation. Now, what I'd like you to note is that even though benzene is bound to this site, which is very far from the active site, it actually induces flap closure. The flaps are not open. The flaps actually remain sort of in a more closed, or maybe, if you want, a semi-open conformation. And this sort of suggests that there must be communication, or there must be some event that's occurring that's transmitted all along this region right up to these flaps. So it could be that the cantilever region is actually affecting the hinge, which then in turn affects the flap tips, and sort of allows those flaps to actually close. So this was the first sort of indication that we should be looking a bit closer to this region over here. So just to show you again, that's the protease, with nothing bound in the active site. The flaps are slightly open or semi-open, but when something is bound, these flaps tend to close in. You would have noticed from the simulation that benzene seemed to touch on this flap pocket over here. It also sampled this site called an exosite, and then it actually preferred to stay for quite a long time in this cantilever site. And that's what we actually started probing. So the cantilever site is sort of at the base of the enzyme. And I'm actually showing in green in the surface representation where the cryptic cantilever pocket is actually located. So this here is the cantilever pocket. Remember, the enzyme active site is over here, and you can see it is actually separated by a reasonably large distance. If memory serves, I think it's definitely over about 15 angstrom. So it is actually quite far away. This was actually quite an interesting result, because when we did this and we looked at the cryptic pocket in the absence of any ligand or any drug, the cryptic pocket would have been sort of, or could have been represented by this green region here. But like I said, it's very difficult to actually observe this pocket when you have an apo form of the protease, or when it's complex to a drug. And this is the situation where it's complex to a protease inhibitor. And the two scenarios are reasonably similar, okay? Although it does appear as if you have slightly more area being shown when a protease inhibitor is bound. But the situation is a lot clearer when one uses benzene. So when benzene, that simple benzene ring, appears in this region, you can see that a lot more of the cryptic pocket now becomes visible. And the same is true for phenol, acetone, imidazole, and isopropanol. Okay, so these simple molecules have actually helped us to reveal a potential site which could not be that visible, or is not that visible in the presence of a protease inhibitor, or even in the apo form. So the cantilever tip, this cantilever region, also changes its conformation in terms of curling in and curling out during these molecular dynamic simulations. And I'll show this in a moment. And the opening of this cryptic pocket actually must therefore be amenable to a certain amount of elasticity, because when it goes in, there are structural changes that are occurring, and those structural changes are obviously favorable because they allow these sort of non-polar hydrophobic molecules to actually bind in this site. So there seems to be some inherent flexibility in that cantilever tip, which gives it this structural elasticity. And this elasticity is also important for the formation of the cryptic pocket. So even though the pocket undergoes this elasticity, the surface of the cryptic pocket is still maintained. It doesn't decompose and becomes unstructured, so to speak. So there is still structure, even though there's elasticity associated with it. So when we wanted to look at the details of the cryptic pocket a bit more, we looked at residues that are within 5 angstrom of each probe. And that's because it's 5 angstrom is, let's say, a good biological cutoff, because most interactions would occur, most relevant interactions would occur within 5 angstrom. Whether they hydrogen bonding, ionic or electrostatic interactions, even these non-specific van der Waals or hydrophobic interactions. So we selected a cutoff of about 5 angstrom. So what we found was under this condition, the cryptic pocket was formed by 4 regions. The one was the NNC terminal beta sheets, NNC terminal beta sheets of the dimer interface, also a certain region of the alpha helix, and then the teens region, so anything from 13 up to 19 of the fulcrum region, also the 60s loop of the cantilever tip. So these 4 regions were actually implicated in the formation of this so-called cryptic pocket, and this is what I'm actually showing on this slide. So shown in the different colors, the diagram on the right, on the left, shows the so-called cryptic site in the absence of any of these molecules, or in the apoform. So you can see it hasn't really formed, it hasn't really shown up, but these are the regions surrounding this cryptic pocket. However, when a molecule is actually bound, okay, these amino acids have actually been found to be within 5 angstrom, and they are contributed by the fulcrum, the active site, the cantilever, the alpha helix, as well as the dimer interface. So there are actually several regions within the protease that contribute towards the formation of this cryptic pocket. And I think that that is quite relevant. So what we did was we used these structure-based software programs, FPocket, DeepSite, CryptoSite, and DarkSite, to try and get some more information about these cryptic pockets. And FPocket identified for us, in most of the simulations, obviously the active site, which would have made sense, but it also highlighted another potential site over here. DeepSite showed us the active site as a potential binding site, but it also identified these sites around the hinge region. And then it also showed us another site around this alpha helix. CryptoSite also showed us binding all the red over here, where binding could occur, and that's around the active site. And it also highlighted the potential site over here, which is the cryptic site. The DarkSite software showed us an additional site in addition to the active site over here on the hinge, the cantilever. It also identified the site shown in blue, which is called the face site. So this cryptic cantilever pocket, which is highlighted by the green arrows, was actually formed by these consensus amino acid sequences. And, a quick visualization of these amino acids would tell us that this pocket is mainly sort of hydrophobic in character. So that means small non-polar hydrophobic molecules could potentially make binding interactions with these residues from these four sites on the protease. So the active site cavity shown in red was the most prominent pocket. F pocket, deep site, and dark site identified two secondary pockets. Okay, two secondary pockets. And deep site and dark site also identify these pockets with an additional site called the face site, which is shown here in blue. And this crypto site, the cryptosite software, showed us that these residues, leucine, aspartic acid, glycine, aspartic acid 29 and 30, as well as these are likely to be part of this pocket. So why is this important? Because if we could engineer a molecule that would maximize the interactions at this pocket with these residues, there is the potential for affecting the enzyme activity of this protease, and I'll get to that in a moment. So surface pockets using structure-based methods. What we did was we used known HIV protease inhibitors. For example, darunavir here is the most recent HIV protease inhibitor. It is highly effective. And you'll see that in cryptosite, F pocket, deep site, and dark site, darunavir has been found to bind in the region that we know it to bind, but it's also located additional sites for darunavir. So darunavir may not only bind to the active site, they also could bind to these other sites. K12 and HIV E9, these are sort of molecules that are undergoing testing at the moment, potential clinical significance, significant clinically. They also identified potential binding sites. So what we actually saw with these programs and these three drugs is that there is a non-active site pocket called the face site, as well as an exosite, a flap pocket, and this other pocket near the cantilever, which is also around the 90s alpha helix. So these are additional sites that could be explored for future use. So the crypto site, this program over here, consistently indicated that the active site residues obviously form part of the pocket, which would make sense. This diagram here, I think, is what sort of really nailed it for us. Because what we did was we took, once we've identified this cryptic pocket, we wanted to know how conserved this pocket is and what the influence of this pocket would be on, let's say, the dynamics of the flaps as well as other regions of the protease. And we looked at the HIV protease, or the protease from several different retroviral proteases. And the first important point here is that the cryptic pocket was always identified in exactly the same location in all of these retroviral proteases. The second is, if one looks at the alpha carbon backbone or the RMSF values, you'll notice that when in the apoprotease for this structure, the flap regions are quite dynamic. As you can see, colored sort of red and yellow, according to this sliding scale over here, with 4 being very flexible, highly mobile, and 3 being quite stable or rigid. So there are quite a few regions in the proteases that are quite flexible. But when you look at the cryptic pocket and binding to this region, you can see how it actually induces folding or stability of the flaps, closing over the active site, and even over here, still quite a bit of flexibility. And even if you look at the T cell, the leukemia protease, you'll notice that there is also quite a bit of flexibility. So, so this actually told us that um, these cantilever regions, um, although they are reasonably dynamic, okay, they can actually induce structural changes in other parts of the protease, and that this behavior is consistent across all proteases. Okay, some to a greater degree than others. So now that we sort of identified the cryptic pocket, its location, we also have an idea of the chemical makeup in terms of the residues. We wanted to ask ourselves whether um, the, this cantilever, this where the cryptic pocket is, whether it actually has an effect on any other region. And so what we did was we looked at generating a disulfide bond, an intramolecular disulfide bond between, oops, sorry, that's cut off. It's residue 72 and 92, and that's from each monomer. So essentially what we've done is we've tried um, to restrict the movement of this or the flexibility within this cryptic pocket by introducing the disulfide bond. And when we did this for the wild type, in the absence of a disulfide bond, we found that, as was to be expected, the flap tips remained in a semi-open conformation with the tips curled out, and you can see we actually measure the angle on the flap tips here about 132 degrees and here 143. So these are called curled out flaps, and these occur when the protease is in the presence of a protease inhibitor. So you can see the flaps are closed, and we can measure the distance between a isoleucine or even a glycine residue on the flap tip and the aspartic acid residue, and that's about 13 angstrom, either way from this monomer to, let's say, the monomer on the left to the aspartic acid on the right, if you will, and the monomer on the right and the aspartic acid on the opposing monomer. So that is what we actually expect with closed flap tips. The situation was different when we introduced the disulfide bond. When we introduced the disulfide bond, the behavior changed in terms of the curling of the tips. Now the tips had actually curled inward, with the angles being reduced from 132 to about 112 and 143 to about 113. So firstly, there was flap tip curling, but in addition to that, the flaps now retained a semi-open conformation with distances of about 19 and 15 angstrom. So clearly, this disulfide bond had actually introduced some structural changes. So stabilization of the cantilever tip via a disulfide bond modifies the behavior of that flap tip. The backbone of the APA wild type and the disulfide bond was stabilized. These root mean square fluctuations ranged from 0.3 to about 4 angstrom, And the wild type actually exhibited a closed flap conformation for about 48% of the simulation, whereas the disulfide bond stabilized structure showed curled in tips and a semi-open conformation for about 42% of the simulation. So what this said to us was that immobilization of the cantilever region using a disulfide bridge could impart conformational changes, and these conformational changes in this region, which are actually felt or experienced by the flap tips, could affect the conformation of the protein, altering its dynamics and potentially its function. Because if the enzyme has either semi-open or closed flap tips, it is highly unlikely or very difficult for the substrate to actually gain entry to this active site. And it might also even affect drug binding. Okay, so clearly, looking at this cantilever and trying to modify or change its behavior can actually affect the structure and the function of the enzyme. So the dynamics can therefore affect the flap dynamics and also the functionality. We then looked at, after these 5 small molecules, we wanted to see if we can identify a larger molecule that could potentially fit into the same cryptic site. So we screened, as I said, just over 60,000 fragments using this general fragment library, and I've just included the site that you can actually access this from. This actually assisted us in predicting binding affinities or the strength of binding, as well as also looking at the mechanism. We only selected the top 1% using very stringent metrics, and we evaluated the top 10 molecules. So what you are seeing here are molecules, different fragments that all bound to the cryptic site, okay, the cantilever site. So all of them bound to the same location, not necessarily in the same orientation. I haven't really shown all the detailed interactions with the other amino acids, but they all certainly show a promise in terms of binding to the site. What I'd like you to just focus on now is just a single fragment, this first fragment, which was our top hit. Okay, when this fragment actually bound to the cantilever site, it induced flap tip closure. Okay, now this type of conformation is only seen, or rather, it's exclusive when there's a substrate that's bound in this active site, or when there is a drug, a protease inhibitor that's bound to the active site. So in this structure, there is nothing bound to the active site. The only molecule bound is now bound to this cryptic site in this cantilever pocket, and it has actually been very effective at inducing flap closure. And this is a comparison just to show you, what a semi-open flap would look like versus a closed flap. Okay. The other important point is that if you look at the RMSF or the flexibility, you'll notice that these flap tips are sort of more red, if you will, in color, which indicates, a bit more dynamic behavior and flexibility. Whereas when the fragment is bound, this region over here does not exhibit any flexibility. In other words, it is very, very stable and it appears to be closed. And this is very good news, because that means that this site most likely would not be accessible to the HIV substrate. So the active site region, as well as the flap tip of the monomer, exhibit lower RMSF compared to the opposing monomer of the fragment-bound complex. What is important here is that all FDA-approved inhibitors, whenever they bind to this site, they induce flap closure. okay? And when the flaps actually so-called lock down, then it inhibits the enzyme and also potentially inhibits its proteolytic activity. So to conclude, what we found was we could identify a novel cryptic cantilever pocket on the surface of an enzyme of HIV protease using this mixed solvent molecular dynamics method. And we didn't use a single probe, we actually used 5 probes. And with each probe, we consistently identified the same pocket. Oh, sorry. We then also, in terms of trying to understand this cryptic pocket, we introduced a disulfide cross-linking, and we saw that there was changes in the flap tip behavior, and also in the ability to change the conformation of those flaps. Um, Changing the conformation or altering the dynamics of that flap or those flaps is very important, because if we can induce closure, we can essentially inhibit the enzyme. Okay? Or if we induce flap opening and cause the flaps to remain open, that could also be a mechanism that one could use for inhibiting the enzyme. Molecular dynamic simulations of our top scoring fragment also identified the cryptic pocket, and that was just further validation for us in terms of the site. And therefore, even this top scoring fragment could be used as a template or a model to generate a library of compounds that could be used to test for enzyme inhibition. The cryptic cantilever pocket, for us, I think represents a novel site that could be explored for drug development. The only limitations of such a study, and I take, I'm fully aware that this is a completely computational study, and we can't go on all of these results as being sort of absolutely correct until we validate them further with some wet lab experimental work. So there are definitely limitations here. So while the mixed solvent molecular dynamic simulations allowed us to identify a pocket, there were certain structure-based programs that allowed us to characterize the features of these pockets. We still have limited or no thermodynamic information regarding the binding and the specifics in terms of the interaction. So in order to do this, there is no doubt that one must do more detailed thermodynamic studies, either using isothermal titration calorimetry, differential scanning calorimetry, SPR, etc., as well as more detailed structural biology studies. And I think Trevor already pointed to this, at the beginning of my talk. So the structural biology is very important if we are to get a much more detailed and comprehensive understanding of the biomolecular interactions at this cryptic site. But I think that the take-home message here is that this cryptic cantilever pocket presents us with a promising target for allosteric modulation. And this is important because we are now able to identify a site other than the active site, or other than the site where a substrate would bind, or even all the current protease inhibitors bind. So looking at allosteric modulation, I think for us is a very exciting and new opportunity to look at how the dynamics of the protease changes and how those dynamics actually impact on function. This work has been published, and it's available in Protein Structure Function and Bioinformatics. And this is work that was done by Dean Sherry and myself. And I just wanted to acknowledge the following people. First and foremost, Dean Sherry, and I've included his picture over here. A really remarkable student. I really wish I could have held on to him longer for a postdoc. We could have done a lot more wet lab experimental work, testing our idea and validating perhaps this hypothesis. Also, my colleague, Dr. Ramesh Pandyan, who generated the molecular dynamics simulations for me. All of our postgrads and our students, my colleagues in the protein structure function research laboratory. We are quite a large laboratory. We have about 40 MSc, PhD, and postdocs. And these students are supervised by myself, Dr. Sylvia Fanuki, Prof. Achilonu, as well as Dr. Ramesh Pandyan. And I really have a fantastic group of people with me. I couldn't have done it without them. So I really must acknowledge all the postgrads, postdocs in our laboratory. Obviously, the NRF for giving us money. And last but not least, definitely Trevor and Chart and all the organizing members. And Trevor, I must say, I really think you're doing a fantastic job with these biophysics online seminars. And I'm really looking forward to seeing this grow much more. Thank you very much.
Thank you very much, Yassine. It was a very clear and interesting lecture. And thank you for the kind things that you said at the end. We really have to, we structural biologists have to stick together. That was what Tom Blundell always used to say.
Yes, absolutely. I agree.
So thanks again. Thank you
Okay.
for the very interesting talk. And I think I'm going to ask people to indicate whether they want to ask questions.
Okay. Thank you. Should I still continue sharing or stop sharing?
Well, I think you might want to go backwards and forwards. So keep.
Okay. Okay. So
Um,
I'll just keep it on the screen. Thank you.
so there's nobody's put up their hand.
I put up my hand.
Oh, okay. Lawrence, go for it. Go for a question.
Yeah, so it strikes me that over the years, there may be a lot of people who have structures that maybe they did not even submit to PDB, where some molecule was in some site that they considered to be off and did not understand what was happening. Have you thought about that, and how do you put, I guess you published a paper, but how do you put the word out that send me whatever structures that you thought were junk, 20 years ago? That's one question. And then the other question was I guess 30 years ago, two molecules that were sort of interesting that were HIV protease inhibitors was buckyballs and benzodiazepine. Did you look at any of those molecules for binding at this cryptic site?
Okay, so thank you for the question, Lawrence. To answer your second question first, no, we did not look at those two. I think that would be interesting. Maybe what I'll do is I could reach out to Dean and just ask him if we can actually use those molecules as well and see if it also exhibits the potential to bind. The first question, yeah, getting the message out there, yeah, that's a bit more difficult because I think people might actually be sitting with structures, as you say, that they've just not deposited into the PDB, and they thought that this is probably not worth exploring. I'm not sure how one actually gets the message out there to them and say, in light of this new information, we actually think you should be re-looking at this. This paper has been published now. I mean, it's sort of reasonably early days. So I'm hoping that maybe if the paper becomes more visible or people start citing it more, that others in the field might actually realize that they could be sitting on potential molecules that could be explored further. Yeah.
Trevor, I have a question. Can I?
Yes, please go ahead.
Yeah, yeah. Thank you for the very interesting presentation, Prof. Said. I just want to ask what effect, whether you were able to run this analysis using HIV protease mutants that are associated with the drug resistance, and I'm also wondering if those conformational changes that you reported could be resolved using maybe simple techniques like limited proteolysis or fluorescence spectrometry, just to mirror the kind of, because I guess what you were showing us are largely just based on in silico analysis. Thank
Yeah.
you.
Yeah. Okay, thanks for the question. So there were two questions. The first is no, we haven't run them on, we haven't done a similar simulations on any of our mutants. This was just sort of exploring this novel idea or concept, and now that we sort of have, if you will, a proof of concept, it's possible that we could definitely run these simulations on many of the mutants that we actually do have in our laboratory. I would just need to find a student who could do that, or maybe I could just speak to Ramesh. He has two high performance computers in his office. And when those computers are idle, I could just step in there and just run these simulations. That shouldn't be difficult. And I think that's important, is to actually compare whether with these mutants, drug-resistant mutants that we actually see coming up in the clinical setting, if these mutants are also capable of still binding or showing this cryptic site, and how the binding of simple molecules or fragments can actually affect the dynamics and potentially the function. So that's something that I think we could explore. Yes, there are other techniques that one could use. Limited proteolysis is one, possibly even doing hydrogen deuterium exchange mass spec. So yes, there are several things. But the only thing is getting a student or students who can actually do this. But I think those, like I said, this is only the computational part, and it's shown us that there is something here. What we need to do now is take that forward into the wet lab scenario, using all these other tools available to us to try and formulate a more complete picture, and in order to see whether this cryptic site is actually a viable site and a target on the protease. But yes, you're right. That's something that needs to be done.
So, Yasin, there are two questions in the chat.
Yes.
The people asking questions would like to ask them. Mohsen, would you like to ask your question? Are you there? Okay, he's gone.
What
No.
was the question, Trevor? Did he indicate
How do
what
you
the question
input
was?
the parameters?
Oh, so you how do you input the parameters? You have to use the software, obviously. So you can either use Schrodinger, or you can use, as we indicated, GROMACS. There are all these other tools, right? So once you're doing the molecular dynamics simulations, if you're opening up the software, you have to actually input, it's like all these different fields that you have, and you have to input the values and make certain changes within the program, and then you run the simulation. So whether you're doing docking or whether you're doing a molecular dynamics simulation, everything is input via, you input it basically via the software. Yeah, so if anybody still needs assistance with that, they're more than welcome to contact me, and we can take them through the process of running let's say, doing a molecular dynamics simulation, or even a molecular docking simulation. Yeah, and I can also refer them to some papers that have come out of our laboratory on methods that one could use for docking as well as dynamic simulations.
That'd be very handy. There is a question that says, will you put your email in the chat?
Okay.
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 And then the next question from Lucas. Why did you use protein structure from the system equilibration and not after the production?
Why did we use? Oh,
Lucas,
okay,
do you want to explain your question?
Yes, I can see it now.
I can ask it. Hello?
Yes,
Go
yes,
ahead.
Lucas. Hi.
Yes. So I was following the presentation. First of all, thank you so much for fantastic presentation. And I was lucky at one of your slide. I noticed that you used the protein from the equilibration, and I was so much interested. Like, why did you didn't use the protein after the production of your MD simulation?
Why did we use the protein structure from the system equilibration and not after the production? Okay, so
Yes.
usually what we do is why we've done that is when you've removed when you take the structure from the PDB, you have to curate that structure in a way where, because remember the structure doesn't have if it's an X-ray crystallography structure, for example, you won't see any hydrogen atoms. You might have some regions that are missing, or amino acids with very strange looking side chains or geometries. So one needs to correct all of that. One of the other things that you need to do is remove your solvent molecules. If you need to, you would remove your ligand or any molecules that are bound, so that you have an apo protein. Taking that protein, you then have to run it through the system so that the structure is equilibrated, so that it actually achieves a stable energy, or the minimum energy required. And at that point in the equilibration, when the structure is not changing significantly, and when the free energy scores are at their lowest, then it's at that point that you would use that structure from the equilibration, okay, in order to run all the other, because that is the most stable structure, and that's why we begin using that protein structure from the equilibration and not just after the production. Does that make sense?
Okay,
So
yeah,
the
sure.
very first step is to always run a system equilibration, so that your structure is stable. Okay, and you can't just use it, from let's say directly from a PDB structure and then run that, because that structure is not necessarily stable. Yeah, so you need to equilibrate the system in terms of all the energies, in terms of the solvent molecules, etc. And once the system has stabilized and it's equilibrated in terms of its energy, then you can select that as your starting point for all the subsequent simulations and docking, etc.
Thank you so much. Thank you.
Okay, thank you, Lucas.
So again, let myself ask a last question, and then you can.
Can I ask a question? I've had my hand up a while, Kev.
I didn't see. Sorry, Wolfie, you go ahead.
Okay, sorry, I didn't want to interrupt, but kind of just a quick question. So do you plan to actually use this information now to develop new inhibitors, or kind of inhibitors coming in from a different angle?
Thanks, Wolf-Dieter. Nice to hear from you. We haven't chatted in a while, but thanks for the question. So I would like to. There there are potentially two challenges there. One is I would need more funding to carry on with the project. And two, obviously I would need a student or students. Currently, my focus has shifted over the past year or so to MPro or the main protease from SARS-CoV-2 and COVID. So I do have all this information. I would like to take it further, but I'm looking at obviously securing more funding for it and then to secure a student.
Just another question in that regard. Have you perhaps kind of checked with people like Frank van Delft, who do these fragment analyses? I mean, it might be that somebody actually has tried to look at this particular molecule, kind of this protease, and has actually verified some of the positions that you found, kind of. And I mean, that might be a quick way of just checking. I mean, not all proteins in the world have been analyzed in this way, but chances for this particular one are fairly high that someone's actually looked at this. And I don't think they publish all of those or kind of put all those structures on the PDB because it's too uncertain.
Yeah, so I haven't contacted Frank, but I think that's a good suggestion. Thanks for bringing that up. I will definitely make a note of that. And secondly, I think maybe in terms of taking this work forward, I'm actually thinking of giving part of this work over to Ramesh, so that maybe he can actually take it and run with it. And because we have access to Diamond, it's quite possible then that we could maybe even order these fragments if they are commercially available, and see if we can potentially, because it's not a problem for us to crystallize the protease. We have the conditions, but it's just to get it as a co-crystal or crystallize it in the presence of this fragment, and then certainly send it over to Diamond. But I think at the same time, once we do have the fragment, or looking at the fragment, I think it might actually just be a good idea immediately to get in touch with Frank and ask him if they've already done something with this, and they already they've got some data already. But yeah, thanks for that suggestion, Wolf-Dieter. I'll definitely make a note of it. Because we do have a list of 10 fragments. So I think I'll pop Frank an email and just ask him if he's found the same fragments or similar fragments.
Yeah, I mean, I don't know whether they've looked at this molecule. Just was something that crossed my mind. Okay, thank you.
Perfect. Good suggestion. Thank you.
Thanks, Wolf-Dieter. I was actually going to ask a very similar question, so I won't ask mine. With that, I'm going to close the meeting. Thank you. Unless somebody else speaks up quickly, there are no other hands up. Okay. Thank you very much for coming. Thank you for listening to Yasin, and thank you very much, Yasin, for a very clear and helpful presentation. Thank you.
Awesome. Thank you, Trevor, and thank you everyone for attending.
Goodbye.
Okay. Goodbye.
Bye-bye.
Thank you.